Morning. Okay. This chapter deals with DNA structure and function, and learning objectives are listed here. We'll go through each one. One of the things we have to appreciate about the DNA molecule is that it's the information storehouse for the tools of life, which are proteins. That's its primary function. Okay. Now, when we talk about DNA, we're talking about a molecule that's built out of building blocks called nucleotides, which have energy built into them because they are very similar to ATP. And we discussed ATP earlier in the course as essentially the energy currency of life, biological gas in the tank, right? So we're going to talk about how DNA holds information, how it's organized, talk about base pairing, and all other aspects of DNA. So we start with a look at some recent developments, right? James Symington brought his police dog tracker from Nova Scotia to a disaster site for the September 11th attacks. He found many survivors who were buried after 3 days of search and rescue. Attacker collapsed from inhalation, burns, and exhaustion. The dog survived but lost his limbs. He died in 2009, but they cloned him. Okay, they made a genetic copy of this particular canine. Soon after Tracker was cloned, canines for police and military forces in Korea began cloning as well. What we have to understand about a clone is that it's a genetic copy of the original organism, sort of like an identical twin. Okay. DNA contains the information needed for the tools of life. Okay. Let's write that down. Okay. Remember that those are proteins. Okay. Let's see a look at Tracker and his clones here. Okay. Notice the similarity, right? Same coat color, same arrangement, everything, right? And we're assuming same built-in ability to follow clues and perform its duties, right? What is DNA for, right? DNA is an information molecule, okay? Meischer first described DNA but didn't know its function. Griffith studied pneumonia-causing bacteria in hopes of making a vaccine and discovered that bacteria that contained a transforming substance that changed harmless bacteria into lethal ones. Okay? We didn't know what this transforming substance was at the time. Turns out it was DNA. Okay. Avery and McCarthy showed DNA was the transforming principle. We'll get into those experiments in just a bit. Okay. For the longest time, right, ever since Mendel, right, we knew that there were heritability factors. Okay. But we didn't know what they were. There were some early candidates. Protein, complex carbohydrates, and dark horse DNA. Okay. Among many other possibilities. Right? But it wasn't until we showed that DNA was the transforming principle that we established that it carried the genetic information, the heritability factors. Okay. What you're looking at is a DNA precipitate. Right there, that white stuff. Okay. This material. In order to function as hereditary material, a molecule has to be able to transmit a full complement of hereditary information to offspring. It has to be found in the same amounts in cells of a given species. It has to be capable of resisting major change, and has to be capable of encoding a large amount of information, enough to build a new organism. Okay. Hershey and Chase confirmed DNA meets the first criteria in 1940 with the bacteriophage experiments we're going to detail here in just a second, using radio-labeled DNA virus. Okay, so let's get this going. Let's pause for a second.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
So the idea behind this experiment was to determine whether protein or DNA was the genetic material. You can radioactively label protein using methionine amino acid with radioactive sulfur attached to it. Okay, and you can manufacture the proteins that make up the coat of the phage, which is a little bit like a virus is to a eukaryotic cell. And you can see whether or not that radioactive signal follows the transformed bacterial cells in the next generation. Right? You can also radioactively label DNA with phosphorus 32, okay? Radioactive phosphorus in nucleotides gets incorporated into the DNA, and the DNA gives off a radioactive signal, all right? And so we can do the same experiment with either hot DNA or hot protein, and where the hot signal goes tells us which is the genetic material, okay? So what happens when you do this is that you end up with, in the case of the radioactive S35, a protein coat that latches on to the outside of the bacterial cell, but with vigorous shaking is dislodged, okay? You would expect if protein were the genetic material that the radioactive S35 would get inside the bacterial cells, but it doesn't, okay? The counterpart to the radioactively labeled protein is the radioactively labeled DNA, right? P32 labeled DNA is radioactive. That, as genetic material, gets injected into the bacterial cell, and that signal is what's followed into the next generation of transformed bacteria, okay? So we have to understand here what's actually going on in this process, right? What is first of all, what is a bacteriophage? Okay, let's address that first. Okay, the bacteriophage is a pathogen of bacteria, meaning it attacks them and hijacks them for its own purposes, which is to make more of these phage. What's it made of? It's made of a protein coat and genetic material. Okay. And what happens when a phage attacks a bacteria is that it lands and attaches to it. That's called adherence. Okay. Attach. Then it injects its DNA or introduces its DNA by fusing with the cell. Okay. And then that DNA takes over the bacterial cell, all right, and turns it into a phage factory. Okay. And the phage produced in this phase of the experiment were radioactive, all right, which means that they had to carry some of the radioactive DNA into the next generation in order to generate new phage. So this becomes the proof, right, that this was in fact the transforming principle, right? This is what changed the bacterial cells into something different, okay? A phage factory instead of a normal bacterial cell. And the phage that were produced as a result of that were also radioactive, okay? So that didn't happen in the case of the S35 labeled phage. They did not carry that radioactive signal into the next generation of phage, and it was not present in the bacterial cells after attachment and injection of the DNA. Okay, so that meant that DNA had to be the candidate. So DNA is the genetic material. Okay. All right. What you're looking at here is an example of the power of DNA. We can take DNA from a donor organism and replace recipient DNA with that DNA, right? So here you can see an example, right? A micropipette punctures the egg and sucks the nucleus out, and this removes all DNA from the egg. Okay. And then another micropipette inserts skin cell DNA from a donor. In this case, the whole cell is put in, an electric current causes the cell to release its nucleus into the egg cytoplasm. If the egg begins to divide, we make a new cell, and that leads to clone of the donor organism. Okay. So that's what we mean by somatic cell nuclear transfer, right? You have all the information necessary to build a copy of the original organism. Okay. You see an example here, right? Cloning cattle by somatic cell nuclear transfer. An example shown here, Champion, a dairy cow. Nelson's estimate Liz on the right, and her clone, Nelson's estimate Liz 2, who was produced by SCNT. Liz 2 started winning championships before she was 1 year old. Okay. And you can see that they are basically copies of each other. All right. Now, you might say, well, wait a minute. They don't look exactly the same. Not exactly the same. Okay. Why is that? And the answer is environment. Okay. There's always two factors that contribute to a trait. One is genetics and one is environment. Okay. Genetics determines the window of a trait. Okay. So you have a ceiling and a floor here, right? And where within that window you fall depends on your interaction with the environment. Okay. Okay. So this is for any trait. Okay. So an example, let's take body weight. All right. You're going to be born with a set of genes that's going to determine the window for your body weight. All right. There will be a ceiling and a floor of weights that you could achieve. All right. If you're outside that ceiling and that floor, we create disease or death. All right. Where within that window you fall depends on what? your diet and your exercise regimen. Okay. So you can see how environment and genetics play a role. Okay. So this is true for virtually every trait. Now, how much of the trait is environment and how much is genetics depends on the trait we're talking about. Right. And the way that you determine the difference between the two is by comparing identical organisms to organisms that are not genetically identical. Okay. And that lets you determine how much of that trait is inherited and what percentage of that trait is a result of environment. So in humans, we would look at identical twins versus fraternal twins. Okay. Identical twins in humans come from a single fertilized egg developing into a cell mass called a blastocyst that eventually splits at some point, and those two cell masses share a placenta and an umbilical cord, and they develop into genetically identical embryos and fetuses, and they are born as the same sex, the same genetic characteristics, the same DNA. Okay. And they look pretty much identical. The differences that you would see in identical twins as they develop would be as a result of only environmental influence, right? Whereas in non-identical twins, okay, the differences that you see are both inherited, genetic, and environmental, all right? So when you look at a trait, if you subtract the difference that's the result of identical twin differences from non-identical twin differences, right? What you get is how much of that trait is genetically determined. Okay. So that would be an example, right? A DNA molecule consists of two strands of nucleotide monomers that run in opposite directions and are coiled into a double helix. The building blocks of DNA, called nucleotides, are composed of a 5-carbon sugar called deoxyribose, 3 phosphate groups, and 1 nitrogen containing base, which is either adenine, thymine, guanine, or cytosine. Okay. And these are all connected to each other by covalent bonds. Okay. So those are the 4 nucleotides of DNA: A, G, C, and T, right? And this is what the building blocks look like, right? There is a base, right? There is a sugar, and there are the 3 phosphate groups, right? They're all attached by covalent bonds. Right? Because they're all nonmetals. Remember, nonmetals in the periodic table. Okay. Why is this called a base? Well, because the nitrogens in this molecule act as proton acceptors. Okay. And as a result, they cause the pH of the solution they're in to go up. Okay. So that's why it's called a base. But the base is really the information part. Of the nucleotide. The sugar and the phosphate is what we would call the backbone, because it's the same in every nucleotide. Okay. So you can see here the different nucleotides, right? A, G, C, and T. Now, if we write these out, right, we would write these as deoxy ATP, deoxy GTP, deoxy CTP, and deoxy TTP. Okay. The energy is already built into each of these building blocks because they each have 3 negatively charged phosphate groups attached to them. Remember that these have negative charges, and so the covalent bonds and hold them together are very, very powerful. And so when those are split, the energy that comes out is used to build the DNA. Okay. Chargaff's rules, which were developed as DNA structure is being investigated, showed that the bases of the two DNA strands in the double helix always pair such that A pairs with T and C pairs with G, but the proportions of A and G vary among species, right? Watson and Crick shared ideas about the structure of DNA and gathered data from other laboratories regarding the structure of DNA, and then theorized what models of DNA may look like from things like scraps of metal. Okay. So they basically brainstormed what the model could be. Okay. They won the Nobel Prize with a one-page paper published in Nature that correctly determined the structure of DNA. Okay. Franklin and Wilkins added that the two double helical strands are held together by hydrogen bonding between the nucleotide bases. Franklin and Wilkins separately worked on identical samples of DNA. But the bottom line is the information got shared and eventually made its way to Watson and Crick, who were able to synthesize it into a working model, right? So all of these people contributed to the elucidating the structure of DNA, right? Chargaff discovered the relationship between the bases. Franklin discovered the structure, discovered a portion of the structure of DNA using X-ray crystallography. Wilkins had experimental evidence of DNA structure, and Watson and Crick synthesized the model, okay? So a lot of these people had pieces of the puzzle, okay? There's a piece. There's a piece. And there's a piece. Okay. Okay. But you have to add them together in order to get the whole model. Okay. Franklin provided critical data about DNA structure, where phosphate backbones were on the outside of the helix and the bases were arranged in an unknown way on the inside. Crick understood the data better and was able to consult an organic chemist that provided the hydrogen bonding that allowed the base pairing to make sense in the context of the model they eventually built and paired according to Chargaff's rules, right? Watson and Crick proposed that DNA consists of two strands of nucleotides running in opposite directions, coiled into a double helix. Hydrogen bonds between the bases hold the two strands together, and there's only two kinds of base pairings that are found in DNA, A to T and G to C, and those were Chargaff's rules, right? So with all of that information together, we ended up with Watson and Crick generating a model from data that came from other sources that was the correct visualization of what DNA is. Now, why do we care so much about DNA? It contains the information used to build the tools of life, which are proteins, and that contributes to the traits of an organism, and that is what allows the organism to function in its environment, okay? So let's take a look at DNA structure.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
So what we have with the DNA molecule is 2 sets of polymers, okay? And the polymers run in opposite directions. So we say that they're what we call anti-parallel. All right, pull that up here. Okay. Anti-parallel. Okay. So the links in the chain of the polymer aren't identical from end to end. Okay. There is one end to the nucleotide and another end to it, right? So with the nucleotides, we know that there's a phosphate end. Okay. And there is a hydroxyl end. Okay. The end that is contains the phosphate is where the energy is at, and that's where the bonds are formed between the nucleotides, right? As we pop off the 2 phosphate groups that are part of the deoxynucleotide triphosphate, the energy that gets released is used to build the chain, okay? So you can see here at the backbone is alternating sugar and phosphates, right? The phosphates here I'm checking in red, the sugars I will check in blue, okay? Right. Sugar, 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 okay, and so on. So the sugar contains the hydroxyl group that allows the next nucleotide triphosphate to attach to it and build the chain in what's called the 5' to 3' direction. Okay. So the 5' end is the phosphate end. Okay. This is the 5' end. And the 3' end is called the hydroxyl end. Right. So that's why we say that the 2 strands run in opposite directions. Okay. You can see in the diagram here that one strand runs 5' to 3', right? And the other strand runs 3' to 5', right? So this would be the 5' end of that strand. Okay. And then up here in the blue, this would be the 3' end. And then the other strand, this would be the 5' end of that strand. And this would be the 3' end. Okay. So anti-parallel. All right. Now, because of that, all right, there's base pairing rules that we talked about earlier, right? A always pairs with T. and G always pairs with C. Okay, those are Chargaff's rules, right? So an A on one strand will pair with a T on the opposite, and a G on one strand will pair with a C on the opposite strand, right? Now, why is that important? Well, the double-stranded nature of DNA and the base pairing rules act like a photocopier. Okay. When DNA gets reproduced, the two strands unwind, and enzymes that make the new strand use the information in the old strand to build the new strand. Okay. And at the end of the process, what you have are two double-stranded DNA molecules, each with one old and one new strand that have the same information as in the original molecule. Okay. So you can see here an example, right, of how the base pairing works. It's just a diagram. It's not really a chemical drawing, right? But that's what we mean by complementarity, right? See how on the bottom strand, we have ACT, CCT, GAG, GAG, right? And on the top strand, we have the complement to that, right? TGA, GGA, CTC, CTC, all right? And so if we were to, again, add a little more information in here, right? This could be the 5' end, and this could be the 3' end. And then on the other strand, let's get, this could be the 3' end, and that would be the 5' end, okay? Again, running in opposite directions, okay? That's important because when you preserve the information, when you reproduce DNA, that allows you to pass it on to the new organism without any changes so that it can look and act like the original organism, all right? That's the key point here. Okay? The DNA in a single human cell would be about 2 meters long if it were stretched out. Chromosome is condensed structure of DNA inside the cell. Twisted and coiled, it offers protection as well as saves space. The histone is spools of proteins that the DNA strand wraps around inside the chromosome. So if we look at this diagram, we can see how the genetic information not only is present as a DNA double helix, right? You can see that here, but that double helix gets wound around itself using these histones as sort of spoolers, all right? And those histones and DNA called chromatin wind around each other, all right? And then when the cell goes to divide, all right, and engage in mitosis, we compress the genetic material even further into a chromosome, all right? And the reason for doing that is to protect the information so that it isn't damaged while mitosis is going on, okay? So when the cell is just doing its work, all right, the DNA is sort of like this, okay? They call that euchromatin. Okay. But when we compress the DNA into chromosomes, our purpose is to protect it so that we can equally distribute it to two new cells, right? With a duplicated chromosome, the information that is here, all right, is identical to the information that is over here, right? That's because we've made an entire copy of the original DNA molecule, right? So there it is. The eukaryotic cell's DNA is divided into a characteristic number of chromosomes. The sum of all chromosomes in a cell or a given type is its chromosome number. The human body has 46 chromosomes. Okay, you get 23 from your father. It's the symbol for dad. Okay, and 23 from your mother. That's the symbol for mom, okay? Male and female, right? Cells having two of each type of chromosome, the characteristic of the species, and that's the diploid condition, all right? Or we could sometimes call that 2N, all right? So 2N equals diploid. Okay. And N equals haploid, right? Okay. What are haploid? Eggs and sperm. What are diploid? Body cells. Karyotyping reveals the characteristics of an individual's chromosomes. The karyotype is an image of the individual's complement of chromosomes arranged by size, length, shape, and location of centromeres. Okay, so let's take a look at how karyotyping is done.
This video, I'm going to talk about karyotypes. First, though, a karyotype will basically show sort of a zoomed out picture of an organism's genome. An organism's genome is its full set of genetic information that it has in its DNA, including all of its genes. And so a karyotype is going to actually be an image of an organism's complete set of chromosomes grouped together in pairs, which we know will be homologous pairs. And they will be arranged in a specific order. We, by convention, order them by decreasing size. And so we number our chromosomes in that way. So you can see we might take out and stain individual chromosomes and then organize them into a picture called a karyotype. This is sort of a nice picture. We looked at this picture when talking about homologous pairs. Notice that we've given each pair a number. So we can talk about chromosome number 3 or chromosome number 12. And they number 1 through 22. Those are going to be autosomes. Autosomes are basically all the chromosomes that aren't sex chromosomes, and then X and Y, or X and X, will be the sex chromosomes. That is our 23rd pair for a total of 23 pairs, 46 chromosomes. This karyotype is of a male because they have, sorry, I didn't mean to cross it out, XY chromosomes. So we should know that XY is male and XX is female. We'll actually look at a couple of exceptions to those in a different video. So this is a female karyotype because it has 2 X chromosomes. So you should be able to look at a karyotype, obviously know that it's a karyotype, and identify if it is male or female.
what you're looking at here is a human karyotype, okay? On the top, you've got a karyotype of a female human with 22 pairs of autosomes and identical sex chromosomes, okay? She is female because she has two X chromosomes, and that's what determines gender. Okay, you can see them right there. All right. And then down here you have the karyotype of an Australian brush turkey. Like other birds, females of the species have non-identical sex chromosomes, ZW. Okay. So sexing is different from species to species, right? But the reason this is important, all right, the first 22 chromosomes here, I'll circle them. Those are called autosomes. Okay. These two down here, these are the sex chromosomes. Okay. The autosomes contain body plan information. Okay. The sex chromosomes determine gender, all right, and some body plan information. On the X chromosome. Okay. You have to have the correct number of chromosomes in an organism at fertilization when sperm meets egg in order to properly build a new organism. All right. If you don't, if you're missing chromosomes or if you have extra chromosomes, then you're going to end up with defects in development that could lead either to abnormalities in physical and mental traits at birth, all right, birth defects, or you could end up in worst case scenario with a spontaneous abortion. Why is that? Because of this idea behind gene dosage. Okay. You have to have the proper gene dosage to build a normal organism. Okay. If you don't have the proper gene dosage, you end up with defects in the process, especially early on, that can be disastrous. Okay. I sort of liken this to building a house from scratch. If you've ever seen a house built from scratch, it's a process, right? You have to buy the land, you have to dig a hole, and in that hole, you have to put a frame made of metal. Around that frame, you have to pour cement to make what's called the foundation. And then you have to, on that foundation, build a frame. On that frame, you have to put exterior walls and insulation and wiring and plumbing and gas, and then interior walls. You have to put in windows and doors, all right? You have to put in the flooring and the roof, and then you have to paint it inside and out. You have to furnish it. You have to turn on the electricity, the gas, and the water, and then you have a functioning house. Okay. Now, if you don't do that in the right order at the right time, you have problems. If, for instance, on the first day of building your house, you ended up with the wrong crew on site. Let's say the painters showed up instead of the people that knew how to pour the foundation, and the foundation was poured crooked. All right. And then that leads to a frame on a crooked foundation and siding on the crooked frame that was built on the crooked foundation, and so on and so on and so on. And you could see that eventually what might happen is that the whole house going to fall over, not going to work. Okay. So early mistakes cost you. All right. And that's what happens when you have an improper number of chromosomes of fertilization. You generate early mistakes. All right. If you say made a mistake on the last few processes of building the house, let's say you didn't finish painting it. All right. The house would probably still function as a house, but it might have some cosmetic defects. Wouldn't be as big a deal. Later mistakes aren't as bad. All right. But with something that's present at the beginning of the process, everything else assumes that the first part of the process is going to work the way that it's supposed to. And if you mess that up, everything else down the road is going to fall apart. Okay. So that's what happens when you have an improper number of chromosomes present at fertilization. Now, when does this happen in the organism? Well, you can have defective eggs or sperm that have improper numbers of chromosomes in them because they don't finish a process called meiosis properly. Okay. In humans, this is generally the result of old eggs. Okay. You're going to find out a little bit later on that in the human, a woman is born with all the eggs she's ever going to have. And then as she ages, her eggs also age. All right. So the older a woman is, the older her eggs are. And the older her eggs are, the less likely they are going to be properly produced, that they're going to be able to finish meiosis properly and have the correct number of chromosomes in them, which would be 23. Right. And if an egg with an unusual chromosome number, an aneuploid gamete is what we call that, aneuploid gamete. The aneuploid gamete is fertilized by a gamete from the opposite sex. You're going to have an aneuploid zygote, and that's going to result in birth defects and or spontaneous abortion. Okay. Generally, in humans, the aneuploidies that make it to live birth are in the smaller chromosomes. Notice that the chromosomes are numbered here in the human from 1 through 22 in order of size. Okay. So 22 is the tiniest and 1 is the biggest. And you might see conditions like trisomy 21, say Down syndrome, trisomy 22. Okay. You might see abnormalities in the number of sex chromosomes. Those make it to live birth, but aneuploidies in the larger autosomes generally are what are called embryonic lethals. Okay. Meaning that they don't even get to the point of making an embryo before the whole thing just stops and it ends up passing through the woman's reproductive system. She doesn't even know it happened. Okay. So this is why karyotyping is important, because karyotyping can tell us about the number of chromosomes in a baby before it's born. And that can indicate to the parents whether or not we have to worry about things like birth defects. Okay. When the cell prepares to divide, it duplicates all of its chromosomes. The sister chromatid is one of 2 attached members of a duplicated eukaryotic chromosome, and the centromere is where the attachment point occurs. Okay. We talked about the autosomes and the sex chromosomes, right? You have one set from your mom, one set from your dad. The set from your mom is called the maternal homologue, right? And the set from your dad is called the paternal homologue. Okay? And you have a pair of each of the chromosomes, 1 through 23. Right? The sex chromosomes differ between male and female. In humans, XY makes male and XX makes female. Okay. So let's look for a minute at DNA replication so that we can understand a little bit more about this process.
In most bacteria, replication of a chromosome begins at a single defined DNA sequence called an origin, or ORIC in E. coli, and ends at a sequence called a terminus. During the first phase of DNA replication, called initiation, enzymes open up a replication bubble at the origin. Enzymes that synthesize DNA can connect nucleotides only in a 5' to 3' direction. That is, every newly made strand begins with a 5' triphosphate and ends with a 3' hydroxyl group. The two parental strands of DNA are antiparallel, which is why the new DNA synthesis will occur in opposite directions. A short RNA molecule, called a primer, is synthesized as a starting point for DNA synthesis. DNA polymerase requires an existing 3' hydroxyl group on which to add nucleotides. But primase, the enzyme that adds a primer, does not. The next phase is elongation. The helix continues to unwind as an enzyme called DNA polymerase III adds DNA nucleotides in the 5' to 3' direction away from the origin. The sites of helix unwinding and new strand synthesis are called replication forks. A circular bacterial chromosome replicates bidirectionally around the chromosome. If polymerases can synthesize DNA only in a 5' to 3' direction, how are both strands of a moving replication fork synthesized simultaneously? At each fork, one strand is synthesized continuously in a 5' to 3' direction toward the fork. The other strand needs to jog back in intervals, each time starting from a new RNA primer. The smoothly elongating strand is called the leading strand, whereas the jogging back strand is called the lagging strand. Note that each replication fork generates a leading strand and a lagging strand, oriented opposite to the other pair. The fragments of DNA that form as a lagging strand is synthesized are called Okazaki fragments. Late in DNA replication, when most of the complementary strand has been elongated, the RNA primers need to be replaced with DNA. The RNA is removed by an enzyme that specifically recognizes RNA-DNA hybrid molecules and cleaves away only the RNA strand. An enzyme called DNA polymerase I fills in DNA nucleotides. The remaining gaps are connected by DNA ligase. Completion of DNA synthesis requires replication at the defined termination or TER site, located on the opposite side of the molecule from the origin. In this termination phase, the DNA helix is completely duplicated and replication stops.
So with DNA replication, the object is to make a new copy of the information so that we can pass it on to a new organism or a new cell, and it can use that information to make the tools of life so it can look and act like the original, okay? Each strand of the double helix is a template for the synthesis of the new complementary strand of DNA, resulting in two new double-stranded DNA molecules, each of which has one old and one new strand. We call that semi-conservative replication. And so you can see that here. During DNA replication, the double helix has to unwind. DNA polymerases uses each strand as a template to assemble the new complementary DNA from nucleotides that it brings in. Each type of DNA polymerase requires a primer in order to initiate DNA synthesis. Okay, DNA ligase will seal the gaps and form a continuous strand once it's finished. Okay. DNA polymerase is a DNA replication enzyme, assembling a new strand of DNA based on the sequence of a DNA template. The primer is a short single strand of DNA that base pairs with a targeted DNA sequence. A DNA ligase is an enzyme that seals the breaks in double-stranded DNA.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
So one strand is made continuously, one strand is made in segments, okay? And the result, unless there is an error, all right, is that the information is faithfully reproduced so that each new organism or each new cell can use it to make the proteins of life that sustain it properly, all right? A mutation is a change in the DNA sequence that comes about because of either mistakes in the enzymes that do the work of reproducing DNA, or interaction with chemicals that change the shape of DNA called mutagens, all right, or damage from radiation, or the interference of viruses that can stick their DNA into the DNA of the host cell, all right? These changes have one of three consequences, all right? They could either be good or bad or neutral, all right? Let's write that in. Good, bad, or neutral. Okay. A good change makes it more likely the organism will live long enough to breed. A bad change makes it less likely the organism will live long enough to breed. And a neutral change has no effect. Okay. And this is what drives a process called evolution and natural selection. Okay. So those are sources of mutation, right? Ionizing radiation, non-ionizing radiation, chemicals, breakdown products, errors in DNA replication itself, right? Is it good? Is it bad? Is it neutral? It depends on where the change takes place and what the change causes, right? Now, if we look at ionizing radiation, you can see damage to these chromosomes in human white blood cells. Very few have the normal shape that you would expect from a duplicated chromosome, right? Where we have, you know, a duplicated chromosome here, its sister chromatids over here. We have more of these elongated structures, okay, or fragmented structures. And that is because of the damage produced by the energy from the ionizing radiation that basically blows chemical bonds apart, right? You can see here an example of mutated flowers from the Chernobyl nuclear disaster, okay? In '86, an accident at the Chernobyl nuclear power plant in Ukraine caused a massive amount of radiation to be released. An example, these flowers were collected in the exclusion zone, an area of about 1,000 square miles. It's still dangerously contaminated. All but one are abnormal, which is an effect of mutation caused by the radiation that was released. Okay. So what you're seeing there is the impact of DNA damage caused by ionizing radiation on the information used to build the tools of life. And so what you end up with are variations in the organism. Now, are these variations good or bad or neutral? It depends on the effect it has on the plant in terms of its ability to survive in its environment. Okay. So not all change is bad. Right? All right. Let's think about for next time advantages and disadvantages of animal cloning. How can karyotypes be used to advise parents or potential parents about their offspring? What would karyotypes not show? And how does a ketogenic diet affect the body? Right? So we'll talk about these in class when we're together next time. All right. I thank everybody for coming, and I hope to see you in class this coming week.